Hello everyone. Welcome to today's video where we will be exploring the world of genetics by extracting DNA from a very common fruit, the banana. Did you know that bananas share almost 60% of their DNA with humans? That's right. DNA is the genetic material that makes us all unique and gives us our traits. In this video, we will be taking you through the step-by-step process of extracting DNA from bananas using simple household items. So, let's dive right in and uncover the secrets of banana DNA. Before we begin, let's make sure we have all the necessary materials for the experiment. You will need a ripe banana, a plastic baggie, a glass, some salt, dish soap, rubbing alcohol, and any type of glass container. Also, the rubbing alcohol, or isopropyl alcohol, should be ice cold, so put it in the freezer around 20 minutes before use. The first step in extracting DNA from a banana is to mash it up. This will help break down the cell walls and release the DNA. Take the banana and place it in the plastic baggie. Then mash it up with your hands until it becomes a pulpy mixture. Next step is to make the extraction solution that will pull out the DNA from the cells. First, I poured 110 milliliters of water into a beaker. Then I added some soap and a teaspoon of salt. Now I poured the extraction solution into the bag with mashed bananas and stirred them around. Then I poured all the liquid in the bag into a beaker. Now the DNA from the bananas is already extracted from its cells, but in order to get pure DNA, we need to isolate it from the rest of the stuff inside this slimy liquid. To do that, I used a colander and poured the liquid in it. This will separate all the unneeded solids in the liquid. This process should take some time, so have patience. After around 30 minutes of filtering, it was done. Now we need to do the final step, and that is to separate the DNA from everything else in this liquid. So I poured about 50 milliliters of isopropyl alcohol, also called rubbing alcohol, and weighted it out. Here's the final result. You can see the slimy DNA getting pulled by my spoon, but I made a mistake during the process and didn't add enough isopropyl alcohol and probably didn't wait enough for the DNA to submerge at the surface. I will try to do better at my next DNA extraction from strawberries next week. See you soon, and don't forget to subscribe.